you administer newborn cells from the animals to them as they aged, the animals who got those cells lived longer than their litter mates that did not get their cells. So many years ago, one of my colleagues and friend and partners, Dr. Craig Venter, the first scientist to sequence the human genome, really led me to understand that he saw our genetic material, our DNA, our chromosomes, our genes, as a programming language. That DNA is like any software language that's used to code so that a computer can follow a set of instructions that are, in fact, there is a set of instructions resident in our DNA that our body and our cells rely on to drive everything from what molecules are synthesized by our cells, what molecules are secreted by our cells into the local environment, and how those cells mature and differentiate or specialize, and what their fate ultimately will be. It's all resident in that genetic material, in that coding language. And I love that. That description, and it made a lot of sense to me. And in fact, if you think about it, the way DNA has a very reliable structure and pattern, it does behave like a true kind of computational coding language. And when I thought about it, I said, well, that's pretty neat. So if our biological software resides in the nucleus of every cell in the DNA, where does that coding language get read? And it obviously gets read in the apparatus, the ribosomes and endoplasmic reticulum in the cytoplasm of our cells, where all of the translation of the transcribed genes takes place in order to produce proteins and peptides and so on. And I said, well, that means that the cytoplasm of the cell is sort of like the insides of the computer. The software resides in the nucleus. The cell is like a compiler, like a computer, and the software gets read. The instructions get passed on. The products from those instructions get produced and either used inside the cell or secreted. So that got me thinking that really every stem cell houses a perfect set of those instructions, a perfect copy of that biological software. And as such, could that stem cell be the best repository or master boot disk if I wanted to reinstall the software, my biological software? So I became very comfortable with a model that basically said that the power of stem cells is that they retain a perfect copy of the biological software, and that biological software was essential to maintaining our health and our functionality as we go through our lives. Now, what's the other important thing to keep note about this biological software? It's written in DNA, the nucleic acid molecules that make up every helix of our chromosomes, every gene, etc. That material is exposed in our lifetime to things like radiation, to things like viruses, to forces that can damage the structure of that DNA, of those nucleic acids, and that damage can be seen as an accumulation of defects in the software. For any of us who remember the days when we had floppy disks and things like that, it was very, very easy to corrupt the software that you were trying to install on your computer. And when you did that, what happened? The program didn't run properly. It crashed. You had errors, etc. So the concept that our biological software was potentially corruptible led me to believe that one of the great potential powers or uses of stem cells would be to keep a perfect copy of our biological software for future reinstallation. That was kind of the way my mind worked. And I got very comfortable that if you thought about every every pluripotent, every versatile stem cell as a master boot disk, and we have the ability today through cryopreservation and so on to collect those cells at birth and store them away, that perfect copy of software gets stored in an uncorrupted, pristine form. So think about this. If your biological software over time in your life, because of viruses, radiation, bad food, poisons, etc., is degraded and corrupted, and that part of the process of keeping you alive is to replace that damaged software. If you had a perfect copy from birth of your software, wouldn't there be an opportune time to reinstall that master disk and bring your software back to its pristine form? So if you think about that as a concept on how to address the diseases and accumulated problems of aging, I mean, anybody who knows how to use a cell phone or a computer is going to figure that that makes a lot of sense. And so that was kind of a standing hypothesis that has led us to explore how to best preserve the quality of the uncorrupted software resident in stem cells, make enough of those stem cells, enough copies of those stem cells that they can be given back to you during your lifetime. And is that a way to maintain your health and the quality of your organs and tissues and their function as we age? And to me, that seems like a very, very logical, straightforward approach that, arguably could be done virtually at any time in your life. So, where we arrived at now is, if you look at the things that we fear most as we age, it's loss of mobility, loss of high performance mobility. If you can't walk around, get around, interact with your environment, that ain't a good thing. Preservation of a high performance cognitive function. You want to be able to think when you're 80 the way you could think when you're 20. High performance immunity. Your immune system cannot accumulate defects and be degraded, because then you can't respond to the threats that are out there, whether they're viruses, pandemic viruses, whether they're damage due to radiation or chemicals. You need to preserve the functionality of that immune system so that it can always be ready to respond to the diseases and threats that we live through. And then, perhaps selfishly, but not too selfishly, preservation of youthful aesthetics, I think, is the fourth pillar of high quality, healthy aging. And so I believe we have tools available today to enable us to preserve that high quality mobility, cognition, immunity, and youthful aesthetics. And those tools ideally come in the form of living cells. And those living cells are the best way to store a perfect master copy of your software that's uncorrupted and can be used to replace anything that gets damaged in your software during your lifetime. So as you can imagine, I'm very interested in the biology of aging and what leads to the degenerative changes that we all want to avoid. And it became very, very clear that the things which lead to the accumulated problems of aging, accumulated mutations in our software, our biological software, degrading the quality of the sort of the machinery of our cells, whether it's our mitochondria, whether it's our protein synthetic apparatus, all that, those things that over time accumulate defects and stop functioning properly is very similar to the things that occur to damage a cell and lead that cell to become cancerous. And so the concept that the biology of aging and the biology of cancer are linked made a lot of sense to me and to my colleagues. And we can break it down into very, very discrete events which we know happen. For example, radiation from the sun that damages the DNA in the cells of your skin that gives rise to a skin cancer like a melanoma. That's an accumulated defect. That is an acquired defect of your biological software that causes that cell not to behave in a normal way and to behave in a malignant way. That's very similar to the fact that the same radiation that caused the mutation that made your skin turn a normal cell into a melanoma cell, that same radiation could damage your cells so that they don't produce the proteins necessary to prevent your skin from wrinkling. So the process of going from I'm in the sun, I get all wrinkled, to I'm in the sun, I've got all these lesions on my skin, it's kind of the same process. And so the fact that the biology of aging and the biology of cancer are so closely linked really suggests that the measures we take to treat cancer and to treat the diseases of aging might use the same tool set. So just to kind of repeat, I said that the hallmarks of healthy aging includes preservation of high performance mobility, high performance cognition, high performance immunity, and youthful aesthetics. Those are the four things I think really matter. It turns out that one of the most obvious signs of aging is what we call frailty. And frailty, kind of loss of strength, loss of flexibility, loss of the ability to protect yourself against damage to your skin, to your organs, etc. All of those things are associated with a decline in specific quality and quantity of cells in your different organs and tissues. The one organ that I pay a lot of attention to is your skeletal muscle tissue. So it turns out that frailty, age-related frailty is a condition called sarcopenia, which simply means loss of muscle. Age-related frailty is characterized by either a complete loss, a loss of the quantity of muscle tissue, or the replacement of healthy muscle tissue with tissues that are not functionally the same. The most common tissue that replaces healthy muscle tissue is either fat or scar tissue. Now, fat and scar tissue don't have the same biologic properties as muscle tissue, aside from the fact that they don't generate force and they don't synthesize the same products, the same proteins and peptides and so on. Fat and scar tissue, not only do they not have that capability, but they actually damage the capability of the residual muscle tissues that's there. If you think about it, if you've got a healthy muscle and it's permeated or it's filled with scar tissue or it's filled with fatty infiltration, that muscle can't possibly function as well as the original muscle tissue. And so preserving muscle seemed to me to be a very actionable strategy to maintain our health as we age. At the very least, it gives you that payback in preservation of high performance mobility. So being interested in muscle and being interested in the concepts of regenerating and building muscle, I looked at all of the approaches, and there are many. Obviously, if you have healthy stem cells inside your muscle and you exercise and you load those muscles, you will actually help preserve them and maintain them and even build them. And you can augment that with nutrition, eating high quality protein, etc. But another strategy would be, okay, if my cells are getting kind of tuckered out in my muscle and they're not doing a great job in replacing lost muscle tissue, maybe if I introduce new muscle cells or new stem cells that can produce muscle, I'll improve the quality of my muscle tissue. So frailty, either related to age or frailty associated with disease, like the frailty that occurs in cancer cachexia, I see this as all targets for regenerative cell therapy to replace, restore, and improve the quality of our muscle tissue. And the net consequence of that, I think, is dramatic. It improves metabolism. It improves immune functionality, and obviously it has benefits both in aesthetics, in strength, and in endurance. So in my mind, that is one of the principal age-related disorders that we can target with regenerative therapies. Several years ago, one of the interesting observations I made was I saw some research done by a scientist named McGonigal from the UK, who measured the number of stem cells in the bone marrow as a function of age. And it shouldn't be a surprise to anybody that as we age, we kind of use up the stem cells in our reservoir that is there to kind of continually renew and renovate our tissues. And that decline was really dramatic in the report by McGonigal in bone marrow. And if I remember correctly, he showed that in the bone marrow of a young child, roughly one in every 20 or 30,000 cells was a stem cell, was actually a stem cell. But when he looked at the bone marrow of an 80-year-old, it was more like 1 in 20 to 30 million. So that's an exponential decline in the number of stem cells in muscle, in our tissues, not just muscle, but in brain and bone, et cetera. It's an exponential decline in the number and quality of stem cells as we age. So the obvious theoretical solution is, well, how do I preserve the number and quality of those cells, and are they replaceable? And so just to kind of test the hypothesis, relatively straightforward, simple experiment is to collect cells, stem cells from the placenta at birth. You cryopreserve them so that you maintain their quality for a period of time, and then thaw and administer them to an aging subject. In this particular case, in aging animals that were part of the experimental design, and then look to see what returning those healthy young newborn cells to an aging animal, what that might do. And the study was a relatively simple, I like to use the term skunk works, kind of a skunk works experiment. And what we found was that if you administer newborn cells from the animals to them as they aged, the animals who got those cells lived longer than their litter mates that did not get their cells. And so that suggested to me that a potential really straightforward approach would be to augment or replenish your stem cell pool, your stem cell reservoir, as we age in order to maintain the function and quality of those important tissues and organs like muscle, like bone, like brain tissue, et cetera. And there's nothing, in my mind, there's nothing simpler than to simply take an off-the-shelf cell product from a newborn, infuse it through through an intravenous catheter, allow it to follow the signals and the mechanisms to tell these cells where to go, and when they get where they're going, they will respond appropriately, and they will mature and specialize into the cells that are necessary for that particular tissue and organ. We've seen it in everywhere from the original work in bone marrow transplantation all the way now to therapies that administer stem cells in order to augment the quality of your skin, kind of rejuvenate your skin and tissues. And by the way, lots of techniques that are used today, even in aesthetics, that call upon the power of stem cells, do so in order to restore functionality, restore or preserve functionality and the anatomic integrity of those organs and tissues. So it's logical, it's intuitively logical, it's a technology that's deployable, and it's an area that we're spending a lot of research and development efforts around. I hope I've given everybody the sense that the placenta is just this treasure trove of stem cells, that you can find very, very high quality stem cells in enormous numbers, and these they're untouched, they're unaltered, uncorrupted from life outside the womb. Well, in our work over the years, we have found that these placental-derived stem cells can basically be used to create any mature cell type. Among the mature cell types that we're interested in are immune cells. And among these immune cells, one of the early ones I got very interested in were parts of the white blood cell system called natural killer cells. So natural killer cells are part of what's called the innate immune system. And the best way to think about it is the innate immune system comes kind of preloaded, knowing what to do. It's pre-programmed, if you will, to perform certain very critical innate functions like defense against common threats that exist early in life. Those threats include things like infectious agents, transmission of cancer, and so on. And we had a very interesting observation that although the incidence of cancer in pregnant women is about 1 in 1,000, transmission of cancer from a pregnant mom to the fetus or placenta is extremely rare, virtually never happens. It's been reported a few times in case reports, although where they don't necessarily link the mother's cancer to the developing cancer in the fetus. It's just more of a coincidence more than anything else. But I was convinced that the placenta actually defends the developing fetus from cancer, defends the developing fetus from infectious diseases like viral infection. And as such, could a placental-derived natural killer cell be an ideal therapeutic tool? Now, it turns out that the way placental natural killer cells identify those threats and destroy them is to recognize certain markers, certain molecular markers on those cells. And among those molecular markers, you have something called a stress antigen. And a stress antigen is a protein that gets expressed on the surface of the cells that say, hey, something's not right here. It's a little bit of a red flag. Now, the expression of stress antigens is a common phenomenon that occurs and can be observed when cells get infected with viruses, when cells are cancerous, they're transformed, and it turns out that stress antigens get expressed on old cells. So old dying cells, what we call senescent cells, also express these stress antigens. So in our work, where we've taken these natural killer cells and have developed them as a therapeutic tool to treat certain cancers because of their intrinsic anti-cancer activity, and we've also worked to develop them as a tool to treat viral infections, kind of generic viral infections. We also recognize that there's an opportunity to use them in the field of age mitigation or anti-aging or longevity by having them clean up the senescent dying older cells that have to be kind of cleaned out of our organs and tissues as we live. The accumulation of senescent cells, for obvious reasons, decreases functionality and the quality of our organs and tissues as we age. So just one example of a cell derived from the population of cells found in the placenta are the immune cells that are part of the innate immune system. The most important one is the natural killer cell, and that natural killer cell can be thought of as kind of a kind of a universal defense tool. And if it can help me protect me from cancer, infectious diseases, and clean up my old senescent cells, imagine how powerful that is as a therapeutic. So I get to hang around a lot of really, really smart people who believe that we're on the threshold of being able to make meaningful progress to delay, arrest, or reverse the degenerative processes that occur in aging. And it's breakthroughs, and we know what some of the breakthroughs are, right? Some of the breakthroughs are eliminate causes of premature death. So using precision medicine to identify diseases before those diseases rear their ugly heads, like cancers, vascular diseases, etc. If you can identify them early enough to intervene, you can prevent that patient from succumbing to that lethal defect. You can also consider that that we have at our fingertips right now tools that can be used to kind of turbocharge our existing systems, like our regenerative systems, like our immune functionality. And one of the, I would say, greatest potential tools to employ is part of the immune system that's designed by nature to protect us from those threats. So I think that the future in aging research and in developing strategies to offset the problems that occur with aging will include cellular tools that use natural immune functions to clean up things, clean up old senescent cells, clean up cells that are infected with viruses, etc. And I think that as a companion to that, we're going to be using more versatile stem cells that have pluripotency, in order to recharge the regenerative engine. So after you get rid of all the bad stuff, you make sure that the subsequent response to disease and injury is done with the highest quality cell possible. And that should be the best way to preserve functionality and quality of our organs and tissues as we age. And that should be accompanied by all of the quality of life benefits that we've been talking about. Being able to move around, think, defend yourself against the threats, and look good.